Hello to everyone. I apologize for the delay. Okay, due to technical reasons, somehow Mac was not very efficient compared to Windows this time. So I have two announcements to give you. So the first one is about the proceedings. I just recalling that we will have a proceedings from this CLAPEM, and the deadline to send your contributions will be on the 30th of April, 2020. So don't worry, we will still send an email just to remind you. And the second one is about the 10th School on Probability and Stochastic Processes, which will be held in Mexico City in August 3rd to the 7th of 2020, okay? So it is my pleasure to introduce Lea Popovic from the University of Concordia, right, in Canada. She's professor there. So since we have a very limited time now, so I think the best is that we start already, right?
Great,
okay.
Thank you. It's an honor to be invited to give a, oops, I should switch this.
Oops, yeah, give me 1 second.
So it's an honor to be invited to give a talk at CLAPEM. It's my first time at such an event in Latin America, and it's been a wonderful conference. And thank you so much for all the organization and the hospitality that you've shown all the participants. I will quickly start by, oops, which way do I go? That way. By explaining what kind of problems I'm interested in working on, describing today. So essentially we're looking at, I'm looking at a problem where there are different types of particles. You can imagine, I'm personally motivated by applications to biology. It's not limited to any specific subset of biology. You can think species types in ecological systems. You can think susceptible, infected, recovered types in epidemiology, or you can even think of genetic types in population genetics. And these particles, these different types interact according to, they have some interactions according to a specific set of mechanisms. So in the epidemiological context, the typical thing would be a susceptible and an infected meat, and potentially the susceptible becomes infected. The rates of interactions will, in my general models, depend both on the number of particles which are close to a certain location and on the specific properties of the spatial location. So because I'm interested in essentially the spatial effects in these models, there are certain interactions that will be able to happen, let's say, only in certain spatial locations, or they will be more prominent in certain spatial locations. So the rates of interactions are dependent not only on the number of types available in that neighborhood, but also on the specific location. And then, of course, since we're in a spatial setting, first I should mention that what I'm interested in typically is that the space is continuous for reasons that have to do with intracellular mechanisms. So in particular, certainly not a lattice or a graph or something like that, because multiple particles should be able to live in on the same, almost the same location. And then the movement of particles is whatever you want it to be, as long as it's Markovian, because those are the tools I know how to handle. So here's the motivation from the biology. Why are the spatial locations? So I particularly am interested in problems that have to do with modeling reactions within cells. And so spatial factors are clearly important because, you know, as you can see from the schematic of a eukaryotic cell, there's all sorts of different locations where things happen. Membrane signaling happens, obviously, on a membrane. certain things happen only in the nucleus, which I will mention, certain things happen only on certain parts of the cytoplasm, etc. So why the spatial factors are very important, because you need certain cofactors to be in the right place for certain mechanisms to happen. Now, why is this reasonable for stochastic models? Is that things like that happen in cells essentially have strong stochastic factors, in particular due to low molecular counts of some key species, right? So in typical ecological problems, or even in epidemiological problems, you can sort of think that most types are in the same amount. Within cells, this is no longer the case. Well, it's debatable whether it's the case in ecology, but good first approximations. But in cells, it's particularly not valid, because you have things that are key, like genes, which are only in one copy, and certain other things like mRNAs in few copies, and other things that are in multiple copies. So the spatial models that have been used so far for intracellular reactions essentially varied from having spatial indeterministics, using reaction diffusion PDEs, to once people sort of were able to capture things on a single cell level, then they started modeling things as spatial and stochastic. And they're single molecular detail kind of models, which I will talk about, and then they're compartment models. So the compartment models seem much more convenient, because you want to say, well, I can just approximate then something with a finite number of locations, and it's a much easier problem to tackle. The problem with that is that the length scales are an issue, because essentially for you to think of a particle, of a system to be homogeneous, it would somehow mean that every particle can get to almost all the other points before, on the order of its lifetime, right? So there's this notion of Kuramoto length, which basically says, how fast do I diffuse? So essentially the distance that I can travel within my lifetime, if that distance is much smaller than the length scale, then you're certainly not any more homogeneous, right? So you can just stay localized in a certain area and then die, right? Before you get a chance to, even if you diffuse, to go somewhere else. So there's that issue of lifetime scaling versus your speed of diffusion. And this schematic kind of exemplifies this idea that if you start, you have to choose this, if you compartmentalize, you have to choose this scale of compartments very carefully. If you choose it very small, such as in this setting, then you end up not having more than one or two particles, and things can't interact. If you bulk them all together, then things die before they're, so it's not an appropriate model. So you're aiming, people who simulate things are aiming for some reasonable in-between scale, which unfortunately, can be correct for some molecular types, not correct for other molecular types, so it's an issue. So anyway, you can there's this sort of asymptotic limits in terms of how one model is kind of a coarse-grained version of another model. So this is just a quick schematic that if you start with a single molecular detail here, and then you diffuse fast, then essentially you will get a certain submodel. Then if you have high concentrations of things, you will eventually get to just an ODE. So there are various, it doesn't, depending on what really happens, fast diffusion, high concentrations, you get different kind of limiting results. And of course, the stochastic effects, what you want to do is you want to still be in the setting where your limit is taken so that the relevant stochastic aspects are still captured. Because obviously, once you get to a one-dimensional deterministic model, then you've probably taken away all of the interesting dynamics that can happen in the phenomenology that's relevant. So let's go to the mathematical framework. So typically, how you would model a stochastic interaction model in this setup is you would set up a vector of counts of different molecular types. So this X is a count of, it's a vector, and the ith component and ID component is telling you how many species of type I in compartment D. So this is in a compartmental model you have. Then essentially you have, this is written in a way where you're driving everything. You're writing a density-dependent Markov chain in terms of time change Poisson processes. So essentially, you run every interaction on a Poisson clock, but its rate is given by these rates that depend on what's going on in that particular compartment. And then, of course, once that, so this kind of counts how many interactions of a certain type K in a compartment D has happened by that time. And then you have to multiply by the change given by these interactions, right? You lose one susceptible, you gain one infected, and that kind of thing. So what's key about not having multiscale aspect, meaning that you have different types in different abundances, is you have to have different scalings of the different coordinates. So you essentially figure out some scaling, you take some generic scaling parameter and you figure out some orders of magnitudes that the different abundances are in, and you scale things appropriately to that. So some things are order 1, some things are order n, some things are somewhere in between. And then you also consider the same thing for the rates, and you get some, multi-scaled Markov chain, right? A quick example of such, so what happens is then you have, depending on what's your renormalized count, some things end up in discrete counts, some things end up in concentrations. And of course, when you take in fluctuations, then certain things fluctuate very slowly once you've renormalized them. Other things fluctuate relative to their amounts quite a lot. So this is a schematic of what happens if you do, I'll be using the gene regulation example in a bit. If you have a gene that can be on and off, and that mechanism happens at a certain rate, but then it has to produce mRNAs, which are in low amounts, which is why you see the fluctuations for the red, right? The gene is the green. And then you have proteins, which are highly abundant, so once you renormalize, then you see very low fluctuations. So essentially the point of this diagram is just to realize that when you look at stochastic dynamics, you have different things happening on different level of fluctuations happening on different time scales. And you want to capture that in some sense in terms of limiting results. Now in compartment models, I did some work a while back because they were simpler, and I sort of wanted to see, okay, if I have a dynamics where some things are, as in this, some things are fluctuating relatively fast and some things are fluctuating relatively slow, if I then enter a rate of movement for these two types of particles, how does the rate of movement affect the final outcomes, given that is the rate of movement of some species important relative to their fluctuations, et cetera. So essentially, if you imagine you have these two different types, like on the previous graph, red and the blue, and you give different speeds to the two different types, then how does that affect the final dynamics, and how do the final distributions of where the movement wants to take them also affect the dynamics? And there's some quick kind of consequences after that I would like to summarize from this theorem, is that essentially for the species in a high concentration, the dynamic depends only on what kind of spatial distribution they have in the long run, right? And it does not depend on how fast they move. Whereas so you have to know what concentration you're in. On the other hand, the other thing that's important is whether you're a fast reacting species or a slow reacting species, meaning whether you have fluctuations on a small time scale. In that case, it really depends on how you move, whether you move faster than your dynamics or you move slower than your dynamics. And it does not depend on where you will distribute yourself, because essentially you will react before you get a chance to distribute yourself according to movement. So these are both intuitively, they make sense. I've recently presented this to biologists, and they all nodded as in, this seems very reasonable, and I did not show them the math theorem. So there's some quick corollaries of this, that then you kind of look at these turnovers in the final nonlinear dynamics, how is that affected by the movement, and you sort of have some quick answers that say, when the fast reaction with the fast fluctuating types also move fast, then they manage to somehow get places to where they need to convert other things before they die. And so the level of nonlinearity is reduced. And in terms of somehow the turnover for the slow species, that really depends on whether you're in some kind of convex or concave turnover situation. And so the spatial heterogeneity can both increase or decrease the turnover there. Okay, now I was not too happy with doing things within compartments, because you essentially are already relying on some division of things, right, into these compartments, which may not really be a good approximation of what you're trying to model. So I went back to sort of modeling things in continuous space, starting from the individual kind of molecular level model. So the easiest way to sort of capture things, right, is you encode all of the different molecular amounts into a measure-valued process with, as many coordinates as you have types. And then you let these measures evolve in time, and they evolve based on both interactions, right? So you have, you have some kind of Markovian movement. They can diffuse, your measure changes because of diffusion, or some of the molecules may stay put. And once certain point masses are within some epsilon distance, you can use some kind of interaction kernel and have them react, right? So the problem is you kind of have to do that around neighborhoods, because whatever, unless you're in 2D, this may not be a good model of interactions, right? So how do you then sort of mathematically sort of analyze these things? Well, you go to martingale problem formulations. So you kind of look at the process on the space of finite point measures. And as I said, you sort of encode everything in point masses. And the only way that you can kind of then recover things, right, is you use test functions, and you use the relevant, if you're looking for averages, you look for the relevant test functions to encode what's happening with the time evolution using the generator of your measure-valued process. You can also, because there are interactions and there's movement, if you have, let's say, diffusive movement and just staying put, then you can also construct this process as sort of a solution to jump SDE, but it's less kind of, that makes more sense if you're doing simulations from this and less from the mathematical analysis point of view. Okay, so what was our goal is, okay, now we have this mathematical framework. How can we exploit different time scales of the dynamics? How can we, in order to reduce the complexity of the model and get some asymptotic result that captures relevant stochastic things? And of course, you're trying to exploit this normalization of amounts, some dynamical scale separation, and some spatial features. Now, of course, there's no single answer to this, because everything depends on what kind of interactions you have and what kind of molecular types you have. So the first thing I did with the student is we looked at a specific, gene expression model, which is very simple. You just have a gene that can be either on because its promoter is activated, or it can be off because its promoter is repressed. Then that gene transcribes into RNA, and then the RNA transcribes into proteins. And then, of course, all of the gene doesn't decay, but the RNA and the proteins just naturally decay. And the localization thing is here very simple because certain things happen. This is the nucleus, the rest is the cytoplasm, the white is the cytoplasm. So in the blue nucleus, that's where the genes sit. So any kind of on and off regulation thing can happen only, right, within, I've coded this blue thing as a ball of radius delta, so it can happen only there. On the other hand, the translation mechanism can in fact happen only in the cytoplasm. So the genes create the mRNA, but the mRNA have to leave before you can create some proteins. And then, in this setting, we'll see that here you will see that the gene is kind of self-regulating because the proteins it's creating will come back to switch the gene off, right? So if you've created a lot of proteins and the protein wants to come back and say enough already, so it comes back and switches the promoter to repressed. Okay, so that's essentially the same thing that I guess I have written down here. So what you have is you have localized species, the genes are localized, the other ones are not, they move, and you have different spatial rates because the regulation happens here, whereas the translation happens only here. All right, next one, I'm just gonna skip. No one wants to see martingale problems in real life. So here two different, we looked at, okay, let's just compare the two simplest kind of settings. Let's look at what we would call the global feedback limit, because essentially you assume that the proteins are fast enough that they can be anywhere at any given time. So it's called global feedback because the proteins from any part of the cell can be quickly in the nucleus and tell the gene whether there's enough rate. So somehow the gene then senses the overall global rate of the proteins in the cell. So that's the assumption that the proteins are fast moving. And so essentially when you use some stochastic averaging results, you get that essentially only the spatial distribution of the proteins matters, how the deterministic spatial distribution that results from its movement. And so you essentially have, you only really care, let's say you're in some the proteins are diffusing, you don't really care about the proportion of the nucleus volume versus the whole cell volume and the total mass of proteins to know at which rate the gene is being regulated to go off versus on, okay? So in this setting, you get a very easy final result because you just get a non-spatial Markov chain for the amount of RNA and for the amount of proteins. You don't need to record anything about space because the protein has moved fast enough that it's just not really important to know where the proteins are being created, and et cetera. Now, in the local feedback limit, the movement of the proteins is not fast, so you essentially have to keep track of where the proteins are. So now the gene is on with a probability that depends on the actual mass of the proteins at locations which are near the nucleus, okay? So what happens is that then as an outcome, your limiting process is still a spatial Markov process because you have to keep track of the mass. So the reason why you have to keep track of the measure value, like the spatial measure value process for the RNAs, they're the ones that create the mass of the protein. So in order to even know whether the gene is on or off, you can write a reduced reaction network, but this factor, which is telling you at which rate you're creating the RNA, essentially depends on the mass, the exact spatial distribution of the mass of the protein. So one thing that's good about this kind of approximation is that you end up with a reduced network, but you still have to, unfortunately, model the spatial Markov process because if you try to take the spatial element out, you will, the process of just the total masses is no longer Markovian, right? That's the part that's inconvenient. So now, just in terms of, what kind of difference does this show relative to in terms of the overall dynamics? Because I somehow claim that if you don't model things properly, you will actually get different phenomenological results out. And it's true that if you simulate things, you see that the local feedback actually shows oscillatory stochastic behavior. So what we're tracking is the number of proteins over time, right? So you can see from, you know, mere 6 simulations that there's this oscillatory stochastic behavior in the local feedback, which is not what shows up if you just simulate the global feedback, because it just wants to go to some steady state equilibrium over time. So it just has a convergence to a steady state, whereas here the oscillations persist over time, and they're essentially due to time delays, because when the protein is created, it takes him some time to come back to the nucleus and regulate the gene, okay, which then regulates how much more new protein you're creating. So the time delay is due to this spatial factor, and then you could say, well, but you know, everyone knows time delay deterministic equations. However, if you write the deterministic approximation for this, the oscillations are attenuating over time. So you actually, they cannot persist, and essentially that's due to the fact that these oscillations that I showed you before, they have a random sort of I mean, I don't know what's called a stochastic oscillation. I mean, they have random periods, they have random amplitudes, and so if you do the deterministic scaling, you will end up with something with attenuating oscillations and not these actual stochastic oscillations. So there are various other coarse grain limits that you could consider here. Wow, I've spoken very fast. My apologies. I assume that people are, thank you for showing up at this early hour. But I'll give you extra time for a break. So there are other coarse grain limits that you can look at even in this small model, which is you can look at what would happen if you had a large number of proteins, which is what in some models is very reasonable. And then what you essentially end up is a concentration scaling of the mass of the proteins, and then it ends up having a similar effect to the global feedback limit, and that ties into the intuition from the compartment models that if you have a high concentration, then high concentration is, because you have so many particles, it's almost like having fast diffusions, because you somehow, with that many particles, you can kind of always have enough around to fill what you need to fill, right? So it has a similar effect to a global feedback limit. Then there are other sort of time scales that are relevant, which is, okay, what happens if the decay rate of the proteins is much faster than the decay rate of the RNA? Then what's interesting, if we go back to this, then what it says is that the mRNA has kind of a longer lifetime, so it leaves, but then the proteins have a very short time, which means that an mRNA then creates a bunch of proteins, and it feels like it's a burst because they're all there for a little while and then they die. And that has like very different, that has a different time scaling, and that we're yet to explore what this bursting does in terms of the regulation, because then you have to scale things so that some of it still can go back and regulate, but then do they do this in some kind of clusters? Do the bursts kind of persist, or how do you take into, or does the burstiness kind of get phased out, right, with the diffusion, and so it smooths over and you don't have any bursts of regulation going on, which is my guess, but we haven't done the full analysis for this yet. Now, what is sort of, in terms of kind of more general theorems that you can do? So we examined this, this specific model, a very simple model. What can you say for very general models? The problem for general models is essentially, if you don't have just linear rates, meaning that you just have births and death creations, right? Which is typically, okay, even in SIR models, you have nonlinear rates, right? But what happens is that you have to make sure that you're somehow controlling the blow up of mass, which even if you somehow say, okay, the reactions are nice enough in a specific like in an overall setting you could still pile up molecules in a certain location so that there you could have blow up of certain kinds. So I guess what I'm trying to say is that you need extra conditions, even if in a homogeneous model, your reactions would behave well and the system would not sort of blow up in terms of counts. That is no longer guaranteed once you're in a spatial setting and things can kind of accumulate places. And so you could have local blow up. So what I'm working on with Amandine Weber are essentially various not too stringent conditions on the types of interaction that allow you to recover. Well, first, you can recover classical scaling limits. When everything is in a large population size, you should get your reaction diffusion PDEs. Again, you have to kind of make these correct assumptions of what happens to the mass, and then you can prove things about uniqueness and regularity. Of course, the regularity is based on a smooth enough diffusion movement of the molecules. And then our idea was like, okay, now we get as a limit some reaction diffusion PDEs, but how can the particles inform us about the properties of these reaction diffusion PDEs, okay? Because that is not an elementary thing either. And then our real interest kind of also lies in what do you do when your normalization is a mix of discrete amounts and continuous amounts? Because this is typically what happens in these multiscale settings. You have, gene of order 1, proteins of order n. So what kind of limit do you get? And we can show that the kind of limits that you get, the kind of stochastic processes that you get are a type, a version of a process that were looked at in some decades back that are called piecewise deterministic Markov processes, which I hadn't heard about until I looked at it. So piecewise deterministic Markov processes are essentially, they're in finite dimensions. You essentially have an x and a y coordinate. An x behaves like a Markov chain whose rates depend on y, and the y just behaves as deterministically according to some ODE, but its ODE drift depends on x, right? So it's a fully coupled Markov chain for one coordinate and ODs for another coordinate. So there's a substantive amount of work has been done on these processes in finite dimensions and how to simulate them well, what are the stability and other properties of these processes. I think they were originally used in queuing, but they have now been applied more to biological things in neural systems, et cetera. However, what we have, because of the spatial component, is we essentially have what I would call an infinite dimensional piecewise deterministic Markov process, because what you have is an x and y component where x is a point mass, so it's a Markov chain on point masses, right? And the y is a PDE. And the PDE coefficients depend on the x, and the various rates for the Markov chain depend on the y. So you have this Markov chain coupled with PDEs, and so you can have all sorts of behavior happening, which is the Markov chain can become a source of waves. So there are all sorts of, there's a whole bulk of, I'm talking a lot about this because I'm trying to enthuse people to take a look at some of these, because I think there's a wealth of various behavior that people haven't necessarily analyzed before from the point of view of, stochastic PDs or anything like that, because it just wasn't a natural kind of model to look at, and in this context it is. So we're looking at questions such as conditions for uniqueness and regularity of the PD element of the limit, and then can you say anything about, long-term behavior, any kind of stationary distribution or something that you can extract. So these are all kind of long-term, what we in Canada like to call long-term project goals. And I will actually finish my talk with this. If anyone's interested in working on these new types of processes, and please get in touch with me. I would be more than happy to have some exchanges and host people and discuss this further. Thank you for your time.
Questions or comments? No? Well, I actually have a question. So all these limiting the objects that you are getting, so I mean, how easy are to handle in order to get some information about the long-term behavior of them, right? So it's
Case by case.
case by case.
Yeah, I mean, like, honestly, I was sort of like, how can I prove that this limit has stochastic oscillatory behavior? And then I sort of was like, well, how's that defined? You kind of have to then case by case say, okay, if I want to claim it has stochastic oscillatory behavior, then I have to kind of define, like how low it goes, like these concepts that they're not, they're not sort of like, they're not generic concepts, and then you can say, okay, this converges to this type of limit. So I think you just kind of have to, there's some hands-on applied mathing field that you have to do in these things. And if it doesn't have complex behavior, then you can say lots of things.
Mm-hmm.
Those are usually not the things that even a biologist is doing. Right?
Mm-hmm.
Steady states is not something, I mean, they want to know things about the steady state, but equilibrium behavior is not the time scale they're interested in. They're interested in the non-equilibrium behavior of these processes. So that's where you kind of have to come up with new tools to describe it. So at least like give them a reduced model or something.
So this self-regulation of protein intensity that you had put up sort of reminded me of like a predator-prey type dynamics almost.
Yeah.
I was wondering if any of the ideas you've developed here, you've thought about sort of applying them to ecological scale systems rather than cellular systems, if you can go from the very small to the very large like that.
Right. Yes, if I see I mean, the behavior is similar, but I think for different reasons somehow. And so I think I don't see direct connections in terms of how you interpret one in terms of the other, but because in the predator-prey models, you have cyclical behavior even in deterministic things, right? Due to conjugates. So unfortunately, there are many versions of things that look the same, right? And they're due to different reasons. So that's the best answer.
Another questions or comments? If it's not the case, then let's thank again Lea for his talk.